Hello, I'm Dr. Mirko Pegoraro, and this is the ninth lesson of developmental biology. It is actually my last lesson. Today we are going to talk about regeneration. Regeneration obviously fascinated humans for a long period of time, mostly because we cannot regenerate much of our organs or tissue. So this fascination with the regeneration ability of some animals obviously infiltrated popular culture, creating powerful heroes or anti-heroes like Deadpool here in this picture or Wolverine. Now, why am I showing you pictures of popular uh, characters or popular heroes or anti-heroes? For in particular, I want you to think about the abilities that Deadpool have in regeneration. So we know that Deadpool can regenerate every part of his body, but actually, if you think about the movies, not each part of the body of Wolverine of Deadpool can regenerate the entire body. If you remember, he cut off his um, um, hand, and the hand cannot regenerate the rest of the body. In fact, it is the rest of the body that regenerates the missing part, which is the hand. Moreover, in one of the movie, Deadpool is cut in half, and only the part which contains the head regenerate the rest of the body, and not the part that contains just the legs. So this suggests that in Deadpool body there is a regeneration signal that controls the regeneration that start from the head and move toward the legs. So there is an anterior posterior gradient, at least of one signal, of one regeneration signal that control the regeneration in Deadpool. And this is very similar to what we will find in some of the animals that we will talk about in this lesson. Probably the same situation is also replicated in Wolverine. However, we never saw in movies Wolverine had removed from the rest of the body, so we are not sure that the same kind of signals are utilized in Wolverine case. So what is the map of this lesson? We will have a small introduction about regeneration in general. Then we will talk about regeneration in three animals, in three groups: Hydra, Planaria, and then we will talk about regeneration in vertebrate, in particular regeneration of the limb in newt. Just to remind you, this morning we had a lesson on metamorphosis, and this is my last lesson, so I will not have another lesson after this one, but I will have a workshop that I strongly advise you to attend because it's a revision workshop. This lesson link very well with the limb development lesson. This morning we talk about metamorphosis, and if you recall, we talk about metamorphosis in vertebrate, amphibian, and as well as in holometabolous insect, like flies. So, different animals have different abilities of regenerate. Hydra and Planaria have enormous ability of regenerate, and they can regenerate entire body from just fragment of the original ones. What is regeneration in general? Regeneration can be defined as the reactivation of development in a post-embryonic life to restore, for example, damaged tissue. So if we cut a planaria in the middle, both parts of planaria will regenerate full individuals. And the same is true for Hydra. Other animals, like frog larvae, regenerate some tissue but not all. For example, frog larvae regenerate the tail, the eye lenses. Zebrafish, which is this fish here, regenerate the tail, the heart, the fin. And newt regenerate tails and limbs. Some other animals, mammals, have reduced regeneration ability. The liver, so most mammals can regenerate the liver, and some mammals can regenerate the antlers. There are four main modes of regenerations. We try to cover them all, more or less, in this lesson. So one mode of regeneration is based on stem cells. So it is a stem cell mediated regeneration. So what are stem cells? Stem cells have the ability to either produce different kind of cell types or they have the ability to produce some kind of cell type. So they can be unipotent or unipotent stem cells, and this unipotent stem cell can regenerate one or two adult different kind of tissues or cell types, or they can be multipotent stem cells, and this can produce different kind of adult cells. So upon damage, a wound, the stem cell reproduce, and they produce, and in reproducing, they also differentiate in different kind of tissue that are missing due to the damage inflicted by the wound. A second mode of regeneration is called epimorphosis. In epimorphosis, the adult cells, they differentiate after damage. So what does it mean? It means that instead of being differentiated in an adult cell type, they become almost similar to stem cells. So they differentiate, so they stop the expression of tissue-specific genes, and they activate the expression of stem cell genes, and then this differentiated cell can re-differentiate in different kind of tissues or one or two different kind of tissues to repair the damage. Morphallaxis. In morphallaxis, which is a kind of repair system that we find in Hydra, the adult cells do not differentiate, but they assume a different adult identity. So they don't differentiate in stem cell, but they become different kind of cells. However, in morphallaxis, we don't have much proliferation, contrary to epimorphosis, where we have a lot of proliferation. And then we have a compensatory regeneration. In compensatory regeneration, the adult cells do not differentiate, do not repattern, so do not change identity. They simply start to cell divide and produce more cell types that are similar to their own or actually identical to their own. And this compensatory system is what mammals adopt to regenerate damage to the liver. So let's talk about Hydra, the ability of Hydra to regenerate, and what kind of expression pattern we find in Hydra that control this ability to regenerate. So first of all, we are talking about the Hydra, small cnidaria freshwater animal, which we see here in this blue picture, and not the Hydra, the evil organization. In the Hydra, the animal, we find a head. In the head, we find the hypostome, which is the mouth and tentacles, and we also have a foot. The foot usually binds to the surface, and it is obviously in water. In the Hydra, we recognize only two layers of cells. So the Hydra is a diploblast, so two layers of cells, an external ectoderm and an internal endoderm. And this structure is called a myoepithelia. Myoepithelia because it has an external epithelium and also cells that function as muscles. It reproduces by asexual reproduction and it produces a new Hydra, new bud, more or less 2/3 from the hypostome, 2/3 down from the hypostome. Now, the body of Hydra is quite unstable. So the cells in the Hydra undergo continuous mitosis and replacement, and as well as displacement. So they are usually produced a lot in the middle of the body, and then they are displaced toward the head, so toward the hypostome or toward the foot. So this means that depending on the age, the different cells will assume different roles. So At one point, one cell will be part of the main body. At one point, that cell will be part of the tentacles or part of the hypostome, and the same cell will be part of the food. So this implies that a specific signal that determines cell fate must be active all the time during the life of the Hydra. Otherwise, when a cell becomes one type, to, for example, become an hypostome, if there is not a signal that keeps telling her what to do, when it moves around, the cell will still be an hypostome, even if now it is a tentacle or a foot. So a signal that specifically tells the cells what to do, depending where they are, must be active all the time in Hydra. Excuse me. The ectodermal and the endodermal cells of the Hydra keep dividing and produce more cells that are similar to the original one. So they produce more ectodermal and endodermal cells. So in general, they produce what are called epithelial cells. But in the Hydra, apart from ectodermal and endodermal cells, we recognize also another kind of cells, which is very important, especially during damage repair, which are called the interstitial stem cells. They're called interstitial stem cells because they live in the middle, in between the different cells of the ectoderm and the endoderm. The Hydra has the ability of Hydra to repair damage is so powerful that if we take an Hydra and we separate all the cells chemically, for example, we separate all the cells, and then we take some of the ectodermal, some of the endodermal, and some interstitial cell, and we put them together in a petri dish, these three types of cells will reorganize themselves to develop a new Hydra. So Hydra is almost immortal, if you want, unless it is eaten. Now, in terms of regeneration ability, we can take an Hydra, cut it in several different pieces along the anterior-posterior axis, and each single piece will produce a new, fully formed individual. In fact, I think one of the most ambitious experiments cut the Hydra in 40 different pieces, which produce 40 different individuals in the end. Not only that, one thing that is remarkable is that each piece will produce the missing part of the Hydra. So, for example, the head part of Hydra will produce a missing foot, the foot part of Hydra will produce a missing head, and the middle part of Hydra will produce both the head and the foot. So the cells in the different part of the animal must have a way to understand what tissue they need to regenerate. So there must be at least an anterior to posterior, if you want, an apical to basal gradient of signals, either one or more signals that instruct the cells and tell them what they need to do in order to generate the correct part, the correct tissue after damage in the right place. So there must be an anterior to posterior morphogen gradient, at least of one morphogen or perhaps multiple morphogens. So, and how do we know that? For example, how do we know whether we are talking about a single morphogen or a multiple morphogens? Well, some of the very early experiments that demonstrate whether one or multiple morphogens are involved are crafting experiments where an hypostome, which is the mouth of the Hydra, have been crafted, moved to a donor in the middle position of the host. When we do this, when the hypostome is crafted on a host, we observe the formation of a new bud at first. So this suggests that there is a signal that from the hypostome that induces the formation of the head. Moreover, when we actually transfer the basal disc, so the basal part of the Hydra, what we have is the formation of a bud that develop basically the basal part of the Hydra first. So this again may suggest that there is one signal, one gradient from the head to the foot. However, when we craft both a piece of the hypostome and the basal disc together in the same place, what we end up having is either no induction at all, or a partial head induction, or a partial foot induction. This particular experiment is conclusively suggesting that we are talking about two signals, two opposite signals. One, head generation, head generating signal, so head activator gradient from the head to the foot, and one foot activator, so a signal that will induce the formation of the foot, the forma gradient from the foot to the head. So we have two signals involved. Now, what are the signals? One signal called the hypostome organizer is a Wnt3 signal. So we find that Wnt3 is expressed in the hypostome, and we can find expression of Wnt3 very early during the formation of the new bud in the reproducing Hydra. And in the hypostome, Wnt3 also induced expression of brachyury. What's important to remember here is that Wnt3 is actually the head organizer in Hydra. And what's happened if we decapitate an Hydra just below the hypostome? The hypostome, again, is the mouth of the animal. Well, if we do that, what we observe is that we have a Wnt3 upregulation on the cell around the wound, and we have a remodeling of the existing cell. So the existing cells over there, they change function to try to compensate for the missing one that are removed by the amputation. And this is a case of morphalactic regeneration. What's happened, however, when we amputate the Hydra in the middle of the body instead of near the hypostome? So if we amputate in the middle of the body, the interstitial stem cells that are around the wound start to die by apoptosis. However, before dying by apoptosis, they increase expression of Wnt3. And this increased expression of Wnt3 will induce an increased expression of beta-catenin in other interstitial stem cells that are around the wound. So this increased beta-catenin expression will induce proliferation of these interstitial stem cells and to repair the wound. So these interstitial stem cells start to reproduce, they start to remodel, so they start to change function, and we have an example here of epimorphic regeneration. So we have morphalaxis regeneration just when the cut is just below the hypostome, where the cell change function but do not proliferate much, while if we cut the Hydra in the middle of the body, the interstitial stem cells start to reproduce, they change function, and they repair the damage, so they basically rebuild up the new part of the new individual. So if Wnt3 is the head signal, what is the foot signal? Well, unfortunately, it is not very clear in Hydra what is this foot activator or this foot signal. One possible candidate for that is Shingard. Shingard, you can see it here in black, is expressed near the bottom of the Hydra, near the foot. More importantly, it is expression just fade away in the place where the new bud during asexual reproduction is produced. And this suggests that possibly Shingard is indeed the foot activator, or if you want, the foot signal. Shingard is also activated by a transcription factor that is called manacol, and manacol is only expressed in the basal disc ectoderm, so in the foot ectoderm. So this again suggests this Shingard may be indeed the foot activator or the foot signal that create this foot to head gradient that control regeneration.